Thank you, Rakesh. You may stop present. Professor Mukhopadhyay, shall we take one more or we'll take a lunch break?
Here we can quickly take one and go our lunch break and again meet at 3.
Okay, okay. Prabir, are you ready?
Yes, sir, I'm ready.
Okay, start.
Please keep the time.
Just
Sorry,
keep
sir?
the time,
Please keep the
exactly
time.
20 minutes.
Okay, sir.
20 minutes and 10 minutes. Okay.
You will get incentive for making it small.
Much shorter, yes.
Okay, sir. Sir, is it visible?
Yes.
Yes, it's visible and you are audible.
Good afternoon, everyone. My topic is functional brain connectivity analysis with brain behavior correlation and EEG-based study under the guidance of Dr. Debashree Guha Adhya. So motivation of my project, a human brain comprises about 100 billion neurons, and it is a very complex in nature, these neurons are anatomically organized over multiple scales of space and functionally interactive over multiple scales of time. So this vast system is the biological hardware from which all our thoughts, feelings and behavior emerge. So there are considerable empirical research from multiple species, theories and mathematical models over the past several decades points to the oscillatory synchronization, sometimes we call it phase synchronization, as being a key mechanism by which neural populations transmit information and form larger network. So we have two objectives. Our first objective is to compute functional brain connectivity analysis using graph theory to investigate hub creation procedure and to test small wall characteristics of the brain as a complex network. And our second objective is we are trying to establish a correlation between the variability of some measure of behavior like button pressing or the variation in reaction time in button pressing with fluctuations in brain waves power in single subject using cross-talk regression. So brain is composed of neurons, and neurons generate some electrical activity, and EEG is a device. So this generation of very small electric fields by the synaptic currents in pyramidal cells, and EEG device used to capture this electrical activity of the brain. So this EEG electrode is not able to capture electrical activity of a single cell, rather than it capture electrical activity of a populations of neurons. So for this, so we have collected two kinds of data: task-based EEG data, resting state EEG data for this thesis. So you can see there is a subject is lying down, and in this way we have captured a resting state EEG data. So we have captured all our data from the neuromedicine department from Calcutta National Medical College Hospital, and this is some stimulus that is shown for the collection of task-based EEG data. So we have sit down the participant on a chair in front of a computer screen, and randomly even and odd numbers are shown in the screen, and the participant is told to press a button whenever there is a even number come. So we have used from RMS EEG India Private Limited a special software to make our raw EEG data that is in .eeg format to be compatible in the MATLAB. So using this RMS EEG software, we are making .eeg file to EDF file, so that we can analyze the data in MATLAB. So this is the console of RMS EEG software that they have given us. So for pre-processing, we use EEG Lab software. So these are the some steps, basic steps for using for the pre-processing of the raw EEG data. So this is my first work, that is functional brain connectivity. So there are three different kinds of connectivity. One is structural connectivity, another using diffusion tensor imaging data, which is anatomical in nature. Then there is functional connectivity, which is symmetric and correlative, and we use EEG data, and our whole thesis is based on this functional connectivity. We are focused only in functional connectivity, and there is another kind of connectivity which is directional in nature. So for that thing, we use functional magnetic resonance imaging, or we can also use EEG. So functional organization of the brain is characterized by segregation and integration of the information being processed. So it is defined as the statistical correlation or the association among two or more anatomically distinct region of the brain. So with a large number of different connectivity methods that are currently available, there is a lack of systematic comparative studies, including a statistical evaluation and their strength and shortcomings of the competing methods. So here is the some intuition of the brain theory behind this phase synchronization. So this is a typical image of an action potential. So this you can see the membrane potential of a neuron is in resting state, but actually it seems to be a straight line, but actually it has some rhythmic feature which is not that much visible in this image, but actually there is some rhythmicity in the brain membrane in the neuron membrane potential. And when the stimulus has come, then some electrochemical events occur in the membrane potential of a neuron, that is depolarization, then repolarization, then hyperpolarization in this refractory period. So this phenomena is called action potential of a neuron, and this plays a very crucial role for transmitting information between the different brain regions. So this is some theory that is proposed by the Fries at 2005. So these three are some population of the neuron. So the idea is like that, from this region, this brain signal is connect. From this population of neuron, this brain signal is coming out, and from this black population of neuron, this brain signal is coming out. And this vertical bar corresponding to these action potentials. So the theory suggests that whenever two brain signals are in phase synchronization, or they have phase synchrony among them, then they exchange information. Because at that time, most of the neural population has in depolarization state, so they are more likely to elicit an action potential. But if two neural population are not in phase synchronization, they are less likely to transfer information among them. So this arrow indicates they are transmitting back and forth the information, but here it is these two brain signals are not in synchrony, so they are not transmitting information between them. So suppose this is a group of neural population, this is another group of neural population, and we have collected EEG brain signal from these two neural population. Now we are interested to find out whether these two electrical brain signal are in phase synchronized or not. So if they are phase synchronized, then they will transfer information between them, and we can call these two neural population as functionally connected. But if they are not in phase synchronized, then we can call that they are not functionally connected. So for this thing, timing, we are more interested in the timing of the signal, not in the amplitude of the signal. So the timing is more important. So this is intertal phase clustering, or ITPC. It is a measure of phase synchronization. So for this thing, we use complex modeled wavelet transform to extract the phase information, also power information in frequency domain. So this is this is the phase angles that we have extracted from complex modeled wavelets from a particular electrode. So here we use PZ electrode, and the same information we are representing in the polar plot after Eulerizing this phase value. So after that, we averaging out these black lines, and we will get one vector, that is this magenta vector. Now the length of this magenta vector corresponds to the strength of this ITPC, or intertal phase clustering connectivity measure. So the same thing we can, so this is the time frequency plot of ITPC using this electrode, and wavelet cycle we vary from 3 to 10, and this color bar indicates the ITPC strength. So in this result we can see when the stimuli has come, after that from 100 millisecond up to this 400 millisecond, there is a strong burst of ITPC, or there is a strong ITPC power we can observe, that is in the from 4 to 8 frequency range, which we can call theta frequency range. So brain power is composed of phase locked power and non-phase locked power. Now phase locked power is those power which are which we can average, which we can make average over the trials. And also event related potential is the trial average of the voltages from all channel. So we can corresponding this phase lock activity of the brain is just the ERP. So ERP is equal to phase lock power, and if we remove the ERP from the total power, we will get non-phase lock power. So here we can see this is the ERP for the standard and the target. Now the standard means when the participant is not pressing the button, And the target means when the target represent when the participant is pressing the button. So this is a typical, this data is taken from particular subject. So for this subject, we can see there is a difference in ERP voltages after the stimulus has come. So this, so we can see clearly visible there is a difference in voltages, and corresponding topo plot we can see also when the participant is pressing the button, his back of this occipital region or this back of his head region is more activated, and there is a less power we can see in the midfrontal area. And this is the total power of this brain region, and we can also see after stimulus has come, there is a high power we can observe here. But if we remove the ERP, as ERP corresponding to the phase locked activity, so if we remove the ERP from the total power, we will get this plot. So this plot corresponds to the non-phase lock activity. So inter-site phase clustering, it is almost similar to inter-trial phase clustering, but the difference is that for this inter-site phase clustering, we are taking electrical brain data or EEG data from the two different channel, and we are just making the phase angle difference from these two channel, and then Eulerizing it. After Eulerizing it, then we make an average. So this vector length depicts the strength of this ISPC. So this is the result from ISPC that is computed between the channel CZ and O1 in task-related EEG data. So these are the two brain signal is in time domain. This is the corresponding polar plot. After Eulerizing, we represent it in the polar domain. And this is the phase angle differences between these two signal that is shown here. So this is the phase angle time series, and it is the corresponding polar plot. So these black lines indicates the phase angle differences from these two channel in polar plot. So after averaging, we get this green vector, which this vector represent inter-site phase clustering measure. So the length of this vector represent the strength of this inter-site phase clustering, and for this particular data we get the result. This length is 0.72949. So this is the strength of this ISPC measure. Then volume conduction, it is a potential artifacts, and the indication of this volume conduction is 0 and pi phase lag. So sometimes what happen, due to this volume conduction, two or more multiple electrodes capture the same electrical activity from the same population of neuron, which can also generate some spurious result in the connectivity measure. So in order to reduce this potential compound or volume conduction, we can use a filter such as surface Laplacian. So this is another phase connectivity method, that is phase lag index or PLI. This measure ignores the 0 and pi phase lag, although its mechanism is almost similar to ISPC and ITPC, but for this measure we only consider imaginary component of the phase differences. So this is the comparison result for between ISPC and PLI. So for this ISPC, it is computed in voltage data, and after applying Laplacian, so we can able to mitigate this potential compound of volume conduction using Laplacian filter. And this for the PLI result, it is almost similar for voltage and Laplacian. So PLI is more robust to the compound of volume conduction. So this is all-to-all channel connectivity matrices. So using complex modular wavelet transform, we can extract phase angle, phase information. Also we can extract power information. Now if we take power value from the two different electrodes and use some statistical correlation like Spearman correlation or Pearson correlation between these two channels, then we can also compute the power correlation between power-based synchronization or power correlation between those two regions of the brain. So this is a comparative study of power-based correlation and phase-based correlation in 3 different frequency range, like 5 Hz corresponding to the theta frequency range, 10 Hz corresponding to the alpha frequency range, 20 Hz corresponding to the beta frequency range. So we can see in this result, for these 3 different brain signals or different frequency range, the corresponding connectivity is also changing. So we can conclude that brain is very much dynamic in nature, and its connectivity method is connectivity is also inside the brain. It is changing very rapidly, and there we can see that phase-based synchronization has almost independent of power-based correlation. So we can conclude that power correlation and phase synchronization are independent in nature. So this data is taken from a resting state subject. So this is the graph theoretical modeling of brain connectivity. So brain is a complex system. So any complex system or network, and this neuron inside the brain compose a network, a brain network. So any network we can better understand using the one mathematical tool, graph theory. So graph theory provides a powerful way of quantitatively describe the topological organization of brain connectivity. So this is typically a graph. These nodes corresponding to the electrodes that capture some brain activity from a specific brain regions, and these edges we can see this is the connectivity strength between these two region of brain or these two electrodes. We can also express graph in a form of a matrix. So this diagonal is black because each channel is auto-correlated with itself. So here the connectivity is 1. So now we are, this is another thing, that is hubness. So hubness is a measure of centrality inside a network. So hub in a network is a node occupying a central position in overall organization of the network. And the hubness is basically a property of a node to act as a hub. So for this thing, so like this here in this graph, we can call A as a hub, and it has the most connection with all other nodes, like 3. So if we remove these A nodes from this network, the whole network or the organization of whole network will be very much disturbed. So to compute hubness, we use some algorithm, like first we compute all-to-all connectivity matrix, then we will make one thresholding. Now this thresholding value, to compute this thresholding value, we take the measure of median of this histogram and plus 1 standard deviation. So using this thresholding, we binarize this connectivity matrix. So above this all value, we made it 1, and below this threshold value, we made it 0. So this is a binarized connectivity matrix. After that, this binarized connectivity matrix, we just make a topo plot for nice visualization. So this is our result. So this upper subplot shows that histogram distribution of phase synchronization strength. So this is the histogram distribution of phase synchronization strength. This is the threshold value. Based on this threshold value, we get this binarized matrix, and this is the all-to-all connectivity matrix at 10 Hz. So we can see this 31 electrodes is a hub because there is a most white part. So white part indicates there is connectivity between all these channels, and this black part indicates there is no connectivity. So those channels which have most of the white part, we can call these channels as a hub. So if we then we converted it in a topo plot, and we can see this region, this whitish region in the brain is more active, or this region is creating a hub for this particular subject. So this is some another measure of hub, as there is some ambiguity in thresholding value. So we can compute this. This threshold value. So this is all-to-all connectivity matrix computed using ISPC, and this shows the topo plot for seeded synchronization. This is the different hubness value for different channels, and this is the topo plot of hubness. This region and this region creating a hub. And to make it more robust or statistically significant threshold, we can see 0.4 up to 0.8, this value corresponding to the significant threshold value. Because less than 0.4, there is a that connectivity is 1. That means all channels are connected to each other, and above 0.8, no channel is connected to no one, because this connectivity strength is 0. So we can see the threshold value we must choose between this 0.4 up to 0.8. So this is the significant threshold. So there is a clustering coefficient. Clustering coefficient measures the connection between neighbor. So suppose this node is a C. So C has 4 connection with 4 other nodes. So we make denominator as 4. And among his neighbors, these 2 nodes and these 2 nodes are connected to each other. So we will make numerator as 2. So this fraction is the measure of clustering coefficient, that how much their neighbors are connected to each other. So this is our result for clustering coefficient. So this is a correlation between clustering coefficient and the degree of connection, and each dot is represented as an electrode. So clustering coefficient and connectivity degree are non-significantly correlated in this data set. This implies that these 2 measurements give separate network organization information, and they are independent in nature. And in this result, we will get that when this clustering coefficient is also threshold dependent. So if we increase the threshold value, this measure of clustering coefficient is also decreases. These all graphs are for different electrodes, and this is the average value of clustering coefficient average over all the electrodes. So small wall network are characterized by high clustering coefficient and lower path length that would be expected by chance, and path length is the average distance from the vertex to any other vertex. Now Watts and Strogatz showed that in the transition between a regular or highly organized network in which neighbors are connected to each other, like this is a regular network where the neighbors are connected to each other, to a random network. This is a random network where the nodes are randomly connected to each other. This path length drops quickly with the increase in rewiring probability. So this is a rewiring probability, whereas clustering coefficient drops more slowly. So this is a small wall network which has some characteristics of regular network, like these nodes are connected to each other neighbor, and also it has some characteristics of random networks, like there is some long distance connection also seen in this network. So our brain, we are going to test whether our brain comprises these small wall characteristics or not. So there is three topology, that is lattice topology where each node is connected to each other, and there is random topology where the nodes are randomly connected to each other, and there is in-between topology, that is complex topology. So complex topology has short length connection, also has long length connection. So here for this thing wiring cost is less, for this thing wiring cost is high. But now we are going to test whether our brain follow these characteristics of small walledness or complex topology. And this rewiring probability means if we remove any edges from a node, then what is the probability of that edge to be connected randomly with another node. So as we increase the probability, rewiring probability, the network become more random. So this is the simulation result based on Watts and Strogatz model. And we can see as we have increases the rewiring probability, so when the rewiring probability is very low, this is almost a regular network, and as we increase the probability, rewiring probability, this network become random in nature, and for in between, this network shows the small walled or the complex network. So and these lines, this orange line indicates the path length which drops quickly as we increase the probability of rewiring, and these blue lines indicates the clustering coefficient. It is also drop as we increase the probability of rewiring. So now we will check whether our real EEG data we can get or we can mimic this result or not, this simulation result or not. So we can see it's significantly able to mimic this result, because as we increase the probability of rewiring, path length is also decreasing, and also clustering coefficient is also decreasing, and so for the low value of rewiring probability, this network is normal in nature. So as we have increasing the probability, it is become more random. So in between, when the probability of rewiring is in between, so we will get a small wall characteristics of the brain. So we can conclude it from this result that our human brain shows the characteristic of small walledness, and this neural and this different neural or the neural population inside the brain make a complex network inside our brain whenever it process any information. And this is the statistical analysis of small wall network. So after permutation testing performed for small walledness, we get a Z value of 16.6711. And in this result, we can see as we have increased the binarizing threshold, the statistical significance of the small wall network is decreases. So as the threshold value increases, the small walledness characteristics of the brain is decreases. So this is our second part of the project, that is brain behavior analysis. So for this thing,
can you go to the results?
Yes, sir. So this is our result for regression between the reaction times and the EEG energy. So these are the variation in reaction times over different trials, and we can see there is a downward trending in the different reaction time. So and this is the beta coefficient for different frequency range. Now the beta coefficient depicts the slope of this regression line. So for this EEG signal at 8 Hz, so this is 8 Hz. For this 8 Hz, we can see it is the beta coefficient is positive. As beta coefficient is a positive indicates the slope. So for this 8 Hz, we get a positive slope. So we can conclude that this reaction time and EEG energy are positively correlated with each other. That means for the larger reaction time, the EEG energy is also high. And for this 20 Hz, we can see for this 20 Hz, this is corresponding to the negative slope. So we can get a negative regression line. So we can conclude that for this 20 Hz, the reaction time and the EEG energy are negatively correlated with each other. So we get from this result a complementary relationship between brain power and the behavior measure at low frequency and at high frequency. And this is the time frequency plot of the regressor, that is beta coefficient, against reaction time over trials. So from this plot, we can conclude that if the reaction time is more, just like between this 500 to up to 1100 millisecond reaction time, so for this high reaction time, we get a high alpha activity, because alpha activity is ranging from 8 Hz to 12 Hz. So we get a value of this indicates a high value of alpha activity that we get for the larger reaction time. And when the reaction time is less, we will get a less alpha activity for this particular behavior. So the conclusion is, we have showed different brain connectivity methods using task-based and resting state EEG data, and we have performed hubness formation inside the brain and the test of small worldness characteristics of the brain that is evaluated and tested successfully. In the second part, a full time frequency map of brain behavior correlation is performed using least squares to do within subject cross-trial linear regression analysis. So in future, we are planning to do a comparative study of functional brain connectivity analysis between normal and epileptic patients, as we have collected lots of epileptic patient data from the hospital, Calcutta National Medical College. So our limitation is graph theory based measure of hubness is threshold dependent. So we have to reduce this ambiguity, and we have to more robust threshold mechanism we have to come out. And another thing is Laplacian spatial filter, although it is a good spatial, it able to make a good spatial separation and mitigate the effect of volume conduction, but it cannot be used everywhere. So we have written one paper, that is functional brain connectivity with hubness using graph theory, that we have submitted in SPCOM, Signal Processing and Communication conference in the IISc Bangalore. And this is the reference, and thank you. And I'm going to just show one small simulation, that is, this is the difference for this difference frequency, how the brain connectivity is changing. So for this brain connectivity is changing. So this is the dynamic, it did capture the dynamic nature, and this is some another one small simulation. So this is the simulation of ISPC, that is, these are the two different brain signals. This is its corresponding polar plot, and this is the phase angle time series. Phase angle difference between these two brain signals are shown in this with the time respect, and this is its corresponding polar plot. And finally, we will compute one average vector from this phase angle vector. So we will get this, and we will get a result 11 between channel 11 and channel 19 is 0.7. Thank you, everyone.
What is the takeaway of your thesis? You have given a lot of data, a lot of analysis.
Sir, the takeaway of this thesis is that we have compared the brain so we have calculated different brain connectivity method, along with we are trying to test the theory that proposed by the Fries, that is the phase-based synchronization. We have tested this thing, and we have successfully able to perform this thing in both in resting state data, because most of the brain connectivity analysis is done based on the resting state data. But we will get a good result also in the task related data. We will get good result, and also we have get something that our brain mimic the behavior of the and the neural network inside our brain follow the behavior, follow the small worldness characteristics, and also the neural network made some complex topology inside our brain. So it might be helpful for some disease like dementia and schizophrenia, where there is a problem with the brain organization. So it will this kind of research we are going to understand more about how this network connection inside our brain, or how brain process information. So this thing, and another is how what is the relationship between the brain and behavior, like some small behavior, how our brain power is changing corresponding to small particular tasks or small behavior.
Apart from that study, is there any application of this information?
Sorry, sir.
Apart from that knowledge gain, is there any other application?
Yes, sir, it has application, like because the neural network that we used in machine learning and all those things, we mimic the neural network from a human brain. So if we understand more that how because human brain is a very complex in nature, and it is the most advanced, we can say most advanced hardware or the most advanced intelligence system that is available to us. So if we know this human intelligence, so to understand human intelligence, this kind of how brain works, this kind of information is required, so that we can make more robust artificial intelligence algorithm or more robust neural network algorithm, and how neural network is forming or interacting with each other. So we can also
have you collected the data?
Yes, sir, I have collected the data from the Calcutta National Medical College Hospital Neuromedicine Department. So they has a separated EEG department. So from there I have collected the data.
Okay, so
and this is the
someone else has actually collected the data.
No, sir. Yeah, they have lots of data. So they have given us data, and we have performed this experiment in that department. So they are able. They are
going
to.
How many subjects you have tried with?
5 subjects, sir. As they have not that much time, so only 5 volunteer I get. So for this, so this is a 3-second duration. So this all data is collected for 10 minutes. So 5 subjects for 10 minutes each. So I have just shown some stimulus inside. They are sitting on a computer, and some number coming in front of that computer, and they are told to press a button whenever there is a.
Another 5 subjects and take the data. Will the results will repeat?
Sir, actually we have tested in these 5 subjects. So that result that we alpha activity. 2 subject we get this changing in alpha activity, but another 1 subject we get that is changing in the theta activity. So I think we need more data to say that which alpha activity or the beta activity or the theta activity responsible for this particular task. So as if now we can I can say that for this particular patient, so I am so that is why I confined this thing in a within subject cross trial regression. So for within subject, for different trial, I am analyzing the data. I am not comparing it with different subject, because each individual brain is different and unique in its nature. So
are you the first person to do this study?
This kind of small cognitive task is that is available. I'm just trying to mimic it, but this number showing and all those.
Compare?
Yes, sir. I can compare, but for this thing I did not compare it. But in future I will compare, sir. And I will try to make.
PhD?
Sorry, sir.
Are you joining PhD?
Yes, sir. I'm planning to do PhD in this EEG and neuroscience domain.
Okay. All the best.
Thank you, sir.
Thank you.
Okay, Dr. Shubhamay?
Yeah, Praveen, good work. So I had one question regarding how do you come up with the hypothesis? So you said that normally we take the resting phase EEG to understand the brain connectivity structure, right?
Yes, sir. But in literature also there are some studies have been done recently, that is they are also find to see how our brain connectivity changes for a different task, and we get some good result for this, like this corresponding to our ground truth, like there should be some brain connectivity changing after the stimulus has come. So we will get that is for 4 to 8 this range of frequency range, there is a high power we can get there is a high power for this small burst. So because brain this kind of burst should be confined within a small millisecond. If it is goes beyond this for 200 to 800, then we can say that this is a spurious result or this is a noise. But as we get small burst of result within a defined that is brain activity, that is theta activity from 4 to 8, so we can I think we can say that this is supporting our ground truth, that there should be some brain activity after stimulus has come. So we can in
Yes,
this way.
that is correct. But the only reason people generally prefer a resting stage EEG is if you have a stimulus, so not the whole brain gets stimulated, but certain pathways get stimulated, which is only one part of the brain,
No,
right?
sir, not only one part. Actually, sir, there is a ambiguity. So whenever some stimulus came inside our brain, so for this thing, there is a human brain project that is going on to because we have a anatomical information of our brain, that this brain is anatomically organized in this way, but actually that information is not able to tell that for how our brain process information. So this is still a mystery. So but it is so for that thing we need some more task-based data or task-based brain connectivity so that we can infer that which regions or what are the portion of the brain that is responsible for that particular task or for that small duration.
Yeah, that you are correct. But that information we can actually get through imaging. So this is what you do with fMRI or any brain
Yes, sir.
stimulation
Yes, sir.
study.
So we
So
can use
in
multiple data like fMRI and other neuroimaging modalities. We can so we can get simultaneous EEG and fMRI. So if I get this kind of facility, then I will also in future I will try to do this thing. So
Okay,
then we will get
so
more.
this brings me to two more questions. So you have multi-channel EEG, so you know which signal is coming from which part of the brain.
Yes.
So how can you use that in your framework to understand? Let's say if you give a visual stimulus, so your visual cortex and
Yes.
amygdala and all this whole entire pathway it gets illuminated. It's
Yes,
not
sir.
just the visual cortex. So this is what you're saying, the related people said that it's not only region, it's not 1 million, but a combination of region. But
Yes.
that is still a subset of the whole brain, right? It's
Yes.
not 100% of the brain. So how can you put pick up this dynamic from the data you have?
Sir, I can say this result like here I show some visual stimulus. So there, so for our back of our head or the occipital region is responsible for our visual information processing. So we can see in this result clearly that this back of his occipital region is more power corresponding to the frontal or mid-frontal region. So we can see this participant is somehow this participant is showing seeing something or some visual stimulus coming inside his brain. So in this way I can confirm that.
And when you also look at the signal, what happens since the signal from this specific pathway is very high, so the resting phase information or the other parts of the brain which is resting, so that goes below sort of your noise level because you have high activity in some
Yes.
other area. So your electrodes are not really detecting this, or like it is detecting, but you cannot take
Actually,
out
sir,
just the,
actually,
yeah.
sir, due to volume conduction, what happened? So suppose one electrode capturing one particular brain activity where we place that electrode, but that is not true. Actually, these electrode captured, so two or multiple electrodes capture the same activity from a same population of neuron. This is a volume conduction which is very much affected this connectivity measure. So for this thing, if we have invasive EEG, then we can more clearly or more robustly we can hypothesize that from which part of the brain we will get this EEG data. Because EEG data we get on the scalp surface, but the scalp surface due to this volume conduction, two or more electrodes can capture the same activity. Now we are saying if these two electrodes are functionally connected, but actually they are capturing the same activity of a same population of neuron. So this result could be spurious in result.
Yeah, and if you look at from the other direction, even if you have an activity map, can you recover the resting phase information for the whole brain from the data? And if you can do it, how will you do the process the signal to give you that resting phase information from activity phase?
Suppose, sir, if we have simultaneously fMRI data, then I can more conclude
Just
that
from the signal, is it possible?
it can be possible using several filters. Like for this thing, I have used this Laplacian filter, and we will get some more good results, some good result that I got using this Laplacian filter. Like this connectivity method is very much contaminated with volume conduction noise, because it shows there is a very high connectivity between these two after this 30 to 40 hertz limit, but actually it is not. So if we apply this Laplacian filter, then we will get this small, because this is the characteristics of brain, that it is activity occurring in a small burst and within a very small millisecond. So after applying Laplacian, we will get this kind of, so this is theta activity, this is around 10 hertz, so this is alpha activity, and this is more than 20 hertz, so we can call this is a beta activity. This information we will not get from this ISPC measure for a raw voltage data. So application of this Laplacian filter, and there are several other special filters that we can also use to mitigate this effect of volume conduction. So from EEG data, in this way we can improve our result. But
Yeah, so you can look at this signal processing methods. I think that will help you go
yes,
to
sir.
the next step.
We can come out with more robust signal processing methods or signal processing procedures, so that we can reduce this kind of noise or reduce this kind of potential confound of volume conduction. Apart from volume conduction, there are so many other artifacts are also present in EEG data, and I have collected it from a raw EEG data, so it is very much contaminated with noise. So for this thing, I have used only small portion of the good portion of the data. So very bad channels and bad portion of the data, I just removed them, so to decrease the size of the data.
And since the data is noisy, how do you set up your baseline measurements?
Sorry, sir?
Like, how do you have the baseline measurements? Because you need to have correct the baseline of the signal when it's very noisy, and then you have to do
Actually, sir, we have actually that resting state data. We have so many resting state data, more than 600. We have resting state data. So from there, I just taken those data, because there are also epileptic patient data. So many epileptic patient data also there. So I have taken only those data which have the ground, because I have also the clinical information I have also got from there. So
that is correct. I'm just saying, when you have the raw signal, how do you do the baseline correction for the signal?
Yeah, for baseline correction, we just follow this thing, that before the stimulus has come, this part I have subtracted. So following some that thing is proposed in a paper of Mike X. Cohen. I have also.
You have not done any of the filtering or signal cleaning steps.
Yes, sir. Yes, sir. I have done many of these things using EEG Lab software.
Okay.
Using like filtering with 0.5 Hz high pass filter, then I remove line noise using notch filter, then I have resampling, and I do average. Also in some data I did not do average referencing, because if the data is noisy.
And why do you use a notch filter here?
To remove the line noise, because line noise, there is a very much noise from the coming out from the electrical line. So this 50 Hz is our Indian in 50 Hz is the line noise. So to remove it, I use a notch filter.
Okay. Okay, thank you. So, Professor Manjuna?
Thank you. Yes, thank you. So any other question from others? Okay, so if there are no questions, Pradeep, you may.
Okay, sir. Thank you. Thank you, everyone.
You may close the
Thank you.
presentation.
Okay, sir.
Professor Sudipto, what time will get back?
Can we get back at the 3?
3. 3 is fine.